Can you see my slide moving from one
Yes.
slide to another?
Yes,
Answer,
doctor.
please.
Yes, doctor.
Yes?
Yes, sir.
Okay. Okay, let me start. Well, this is our topic for today, transcription and regulation of gene expression. So what is transcription? Well, it is the biosynthesis of RNA, wherein RNA copies of selected DNA sequences are made. And by this DNA sequences and selected at that, I am referring to the gene. And the gene is made up of DNA, and genes are present in the chromosomes that are present in the nucleus. So this is how we define a gene. It is a segment of DNA that codes for an RNA, a polypeptide, or protein molecule. Now, this is the molecular definition of a gene. And the three major products of gene transcription are messenger RNA, transfer RNA, and ribosomal RNA. So transcription is the first step in gene expression, in which information from a gene is used to construct a functional product such as a protein. And the goal of transcription is to make an RNA copy of a gene's DNA sequence. The original genetic code is present in the DNA, but the DNA cannot be a direct template for the biosynthesis of a polypeptide or protein. As I have explained to you the last time around, the flow of genetic information is one way, and that is from DNA to RNA and then to protein molecules. For a protein coding gene product, and that is the messenger RNA, it carries the information that is needed in order to synthesize a polypeptide or even a subunit of an oligomeric protein. Now, eukaryotic RNA transcripts need to go through some post-transcriptional processing steps before translation into proteins. And we're going to study that post-transcriptional modification in our lecture. Now, these are the similarities and differences between replication and transcription. First, the similarities. Both of them are present in the nucleus, and they require a DNA template in order to synthesize a DNA and in order to synthesize an RNA. The difference is that in replication, the entire parts of the DNA are copied, while in transcription, only segments of the DNA are copied. And what are those segments? Those are the genes, okay, that are present in the chromosome. In replication, the two strands of the DNA template are replicated, while in transcription, only one of the two strands is copied into an RNA. You have learned that in replication, it requires an RNA primer, but that is not so in transcription. There is no RNA primer that is needed because what you are synthesizing is already RNA. The major enzyme in replication is DNA polymerase, while in transcription, RNA polymerase. The similarity is that both these polymerases form phosphodiester bridges between nucleotide residues. Another similarity is that the direction of elongation of the daughter DNA and of the RNA transcript is from 5' to 3' direction. The difference in the nucleoside triphosphates required is that in replication, you need deoxynucleoside triphosphates like DATP, deoxyguanosine triphosphate, deoxycytidine triphosphate, and deoxythymidine triphosphate. But during the condensation, as I've lectured to you before, the pyrophosphate is removed. So what actually condenses are the deoxynucleoside monophosphate, DAMP, DGMP, and so on and so forth. Now, the same thing is true in transcription. Okay? Only in transcription, what we need is the ribonucleotide of uracil and not thymine. And the sugars is ribose. Okay? So this is the classical definition of a gene. It is a segment of DNA that determines a single character or phenotype of the organism. And what is a phenotype? Well, that is the observable characteristic of the organism, like this one, eye color, height, and so on and so forth. Now, an organism's genotype is the set of genes that it carries. So the genotype consists of the entire DNA that is present in the nucleus. An organism's phenotype is all of its observable characteristics, which in turn are influenced both by its genotype and also by the environment. For example, differences in the genotypes can produce different phenotypes. And these are examples of phenotypic characteristic of man and other mammals. Now, phenotypes also include observable characteristics that can be measured in the laboratory. So if you request for a complete blood count and the results come in, then the number of red blood cells, the number of white blood cells, that can be considered as a phenotype. Okay? But we are more interested in the molecular definition of a gene. It is a segment of DNA that codes for a protein, a polypeptide, an enzyme, and an RNA. Okay? And why enzyme? Because generally speaking, enzymes are protein in nature. So what are the types of gene from the standpoint of function? Well, we have structural genes that codes for polypeptides and RNAs. And then we have non-structural genes, such as operator gene, one that serves as a starting point for reading the genetic code, and the regulator gene, one that synthesizes a repressor, especially in the prokaryotes. A repressor is a substance that switches off the activity of the structural gene. So a structural gene is a gene that codes for any RNA or protein product other than a regulatory proteins. Okay? So they do not code for a structural gene, does not code for regulatory proteins that are involved in the regulation of gene expression. Structural gene products include enzymes and structural proteins. Also encoded by structural genes are the non-coding RNAs, such as the ribosomal RNA and the transfer RNA. Okay? So the transcription, the initiation of transcription begins the process of transcribing the DNA message of one or more structural genes into messenger RNA. So the structural genes are linked to an operator gene and a functional unit called an operon. And this is particularly true among prokaryotes or bacterium, but not so in mammalian cells. Okay? So the operon system is not present in our body. Okay? Or if ever it is present, it is not well developed as in prokaryotes. So regulatory gene is a gene involved in controlling the expression of one or more other genes. Okay? So that is the difference between the structural gene and the regulator gene. And the correct word there is control. Controlling of gene expression. In prokaryotes, the regulator gene often code for a repressor protein. And we're going to study this later on in lock-I operon. Okay, in lock operon, I mean. Repressor proteins bind to operators or promoter site of the gene, preventing the RNA polymerase from transcribing the DNA into an RNA. So these are the other terms that you have to be familiar with, exons and introns. Exons are coding segments of the gene, and they contain the codons that specify the kind of amino acids that will be incorporated in a protein molecule during translation. Introns, on the other hand, are non-coding segments of the gene. There are no codons in the introns, but they are involved in the regulation of gene expression. Now, introns and exons are nucleotide sequences within a gene. Okay? So genes contain introns and exons alike. Okay? Now, later, introns are removed by RNA splicing as RNA matures. This means to say that introns are not expressed in the final messenger RNA product. That means to say that the mature messenger RNA does not contain introns anymore. they only contain exons. So these exons become covalently bonded to one another in order to create a mature messenger RNA. And what's that bond? Phosphodiester bond. So I repeat, exons are coding areas of the gene. Introns are not non-coding areas of the gene. Now, furthermore, exons are interspersed with introns in most eukaryotic genes. Okay? They go together. Okay? There are some areas where you have exons, other areas where you have introns. And the entire gene is first copied into a pre-messenger RNA that we call hnRNA. HN is short for heterogeneous RNA during transcription. And what is the heterogeneity all about? The presence of exons and introns in the hnRNA. Okay? Next, during the process of RNA splicing, introns are removed. I mentioned that already. And exons are joined together. Okay? So what happens to hnRNA? It becomes a mature messenger RNA ready for translation after the removal of introns. In molecular biology, RNA splicing is a form of RNA processing in which a newly synthesized precursor messenger RNA is transformed into a messenger RNA. Okay? So during splicing, the non-coding regions, what are they again? Introns. They are removed. And the coding regions, again, what are they? Exons. They are joined together. So these are the different kinds of RNAs that are produced during transcription. First, we have the messenger RNA. It contains the codons that specifies the sequence of amino acid in a protein. That's why it is called protein coding. The non-protein coding RNAs are transfer RNA. It contains the anticodon that matches the codons present in the messenger RNA. It also serves as the carrier of the amino acid at the site of protein synthesis, which is the ribosome. Then the third major RNA product is ribosomal RNA. It becomes a part of the ribosome. So this ribosomal RNA is actually a ribozyme. Okay? It has a catalytic activity. And during translation, this ribosomal RNA becomes your peptidyl transferase. Then you have the long non-coding RNA, and it is involved in chromatin remodeling. Then small nuclear RNA involved in the removal of introns and splicing of exons. So this is the one involved in the conversion of heterogeneous RNA into a mature messenger RNA. And then you have the microRNA and the silencing RNA that modulates, regulates gene expression. Okay? So these are the three major products of transcription. So the messenger RNA is a single-stranded RNA molecule that corresponds to the genetic sequence of a gene, and it is read by the ribosome during the process of translation or protein synthesis. So messenger RNA carries the genetic information copied from the DNA in the form of a series of three base code or words that we call codon. And each codon specifies a specific or particular amino acid. Now, what about the transfer RNA? Okay, sometimes it is also called soluble RNA or sRNA. It is an adapter molecule composed of about 76 to 90 nucleotide residues. And it serves as a physical link between the messenger RNA and the amino acid that it carries. Remember, tRNA is the carrier of the amino acid. Okay? And what do you have in the transfer RNA? Anti-codon that matches the codon in the messenger RNA. Okay? And this is what I told you. tRNA carry the amino acid to the site of protein synthesis, which is the ribosome. Okay? And it is directed by a codon or codons in the messenger RNA. So transfer RNA are necessary components of translation, the biological synthesis of new proteins. And that is guided or in accordance with the genetic code. Okay? Its genetic code is made up of three nucleotide residues which make up the codon. Okay? I'm referring to the codon here. Ribosomal RNA, on the other hand, is a type of non-coding RNA similar to your transfer RNA, and it is a primary component of the ribosome. It is a ribozyme that carries out protein synthesis in the ribosome. And what ribozyme am I talking about? Peptidyl transferase. Peptidyl transferase. So let's talk about DNA-dependent RNA polymerase. It is the principal enzyme involved in transcription. It is the counterpart of DNA polymerase in replication. And one thing that makes RNA polymerase differ from DNA polymerase is that it is involved in all steps of transcription. Recognition of the promoter site of the gene, initiation of RNA synthesis, elongation of the RNA transcript, and termination of transcription. Now, the RNA holoenzyme consists of the 2 alpha subunits, 2 beta subunit, 1 omega, and 1 sigma subunit. So it is a hexamer. 2 plus 2 plus 2, 6. It is a hexamer made up of 6 subunits. Now, why do we call it DNA dependent? Because this RNA polymerase requires a DNA template in order to synthesize an RNA. Okay? It is an enzyme that is responsible for copying a DNA sequence into an RNA sequence during transcription. RNA polymerase locally opens the double-stranded DNA, about 4 turns of the double helix, so that one strand of the exposed nucleotides can be used as a template for the synthesis of RNA. So in other words, RNA polymerase is acting like a helicase. Remember what helicase is? It is an enzyme that will unwind short segments of DNA. Here, RNAP can also do that. It can open, okay, the double-stranded DNA, okay, so as to separate the 2 strands. But only one of the 2 strands of the DNA is transcribed. Okay? So RNAP not only initiates RNA transcription, it also guides the nucleotides into position, then facilitate attachment and elongation, okay, has intrinsic proofreading and replacement capabilities, and termination recognition capability. Okay? So what is the core enzyme made up of? Well, it is a 2 alpha subunit, 2 beta subunits. I mentioned this. These 2 betas are beta and beta prime, 1 omega subunit. Okay? The sigma subunit is a regulatory subunit. It recruits or helps the core enzyme to attach more tightly to a specific site in the promoter region of the gene. Okay? So in prokaryotes, there's usually only one kind of RNA polymerases. In some instances, they are multi-subunit, like this one. In some species, it is only a single, okay, single subunit RNA polymerase. Now, we are now interested in our body, okay? What are the different kinds of RNA polymerases that we have? RNA polymerase 1. It synthesizes the ribosomal RNA. And it is not inhibited by amanitin. Amanitin is a toxic principle found in poisonous mushroom. Then we have RNAP2. Okay? This is the one that is inhibited by amanitin. Okay? When the poison is, even if the poison is present in low concentration. Okay? RNAP2 synthesizes the heterogeneous nuclear RNA that contains the introns and exons. Okay? It is the precursor of the mature messenger RNA. RNAP3 is not inhibited by amanitin. It synthesizes the transfer RNA. Okay? So initiation of transcription is a primary means for controlling gene expression. In the bacteria, the RNA polymerase holoenzyme binds and unwinds promoter DNA, forming the transcription bubble of the open promoter complex. Okay? Just like your replication bubble, just like your replication eye. Okay? A core enzyme consists of the subunits of an enzyme that are needed for catalytic activity. Okay? And these are the core enzymes for most of the prokaryotes. Okay? So this is the replication, I'm sorry, this is the transcription bubble that I'm talking about. And that is produced by the RNA polymerase complex. And once the double-stranded DNA opens up, only one of the two strand is transcribed, this one. Okay? And it is copied. And this is your RNA transcript. Okay? So let's now go to the basic steps in transcription. First is template binding. Recognition of a specific binding site called promoters in the DNA template. What are the characteristics of the promoter site? Well, it is an AT-rich region. Okay? Just like the replication bubble, just like your ORI site. Okay? The reason is the same. Okay? AT-rich regions are easier to unwind, and they are targets of regulatory effectors. So regulation of gene transcription happens at the level of the promoter site of the gene. Okay? So these regulatory substances called effectors or modulators, okay, they either stimulate gene transcription or inhibit gene transcription. And look at this one. The sigma subunit enhances the binding of the core RNA polymerase to this promoter site of the gene. Okay? So, what are the characteristics of the promoter sites of the gene? Okay? Now, promoters contain regulatory sequences such as response elements. We call them response elements that provide secure initial binding for RNA polymerase. Okay? And for proteins called transcription factors. So, what kind of proteins bind to the promoter site? RNA polymerase, the major enzyme itself, and transcription factors. Okay? Now, these transcription factors are proteins. Okay? They have a specific activator or repressor sequences of corresponding nucleotides that attach to specific promoters and regulate gene expression. And that's the reason why I said earlier that this transcription factor, once the modulator or effectors bind to them, that may result in stimulation of transcription or repression, inhibition of transcription. In bacteria, the promoter is recognized by the RNA polymerase and an associated sigma factor. On the other hand, in eukaryotes, the process is more complicated, and at least 7 different factors are necessary for binding of an RNA polymerase 2 to the promoter region of the gene. Transcription factors. Okay? Okay. What do we have here? Okay, we have here a segment of the DNA. And in this segment of DNA, we have 4 genes. A, B, C, and D. Okay? And you will note that in there are your DNA template. Okay? It is made up of the DNA. Remember the definition? A gene is a segment of DNA. And there are two kinds of strands, the template strand and the coding strand. Let us take up first the template strand. You have to know the other names because in the examination question, I may be using the other names, non-coding and antisense. So the template strand is the DNA strand that is transcribed into an RNA. And in your illustration, they are the shaded strand. Okay? So the template can be located in any one of the two strands. So genes A, B, and D, they are found in the lower strand. While in gene C, the template strand is the upper strand of the duplex. And look at the arrow. Okay? It runs in 3' to 5' direction because that is how RNA polymerase reads the base sequence of the DNA template, 3' to 5' direction. Look at gene C, same. Look at the arrow. From 3' arrow to 5' direction. Okay? The coding or sense strand, on the other hand, is the untranscribed DNA strand. And it contains the regulatory elements. Okay? The regulatory elements are here. Okay? In the coding strands. It contains the regulatory signals. So this is an illustration of a prokaryotic gene, and this is the transcription start site. Okay? And that is where transcription begins. The nucleotide from here up to there is given a positive number. Okay? While the region to the left of the transcription start site is the promoter site. So this is the promoter site. Upstream of the transcription start site, the transcribed region is downstream. Okay? This is the one that will be transcribed into an RNA. This one. Okay? And in prokaryotes, you have the so-called Pribnow box or TATAAT box. Remember that. And what is the importance of this TATAAT box or Pribnow box? It tells the RNA polymerase where exactly is the transcription start site. And that's the reason why TATA box or Pribnow box is also called the -10 region. Okay, because all nucleotides to the left of the transcription start site is given a negative number. Okay, so the TATA box will tell the RNA polymerase 10 bases away from me is the transcription start site. Okay, so it is a sequence, the Pribnow box. It is a sequence of thymine, adenine, and thymine. 6 nucleotide residues. And it is an essential part of the promoter site on the DNA for transcription to occur in bacteria. It is an idealized or consensus sequence, okay, that shows the most frequently occurring base in many promoters that have been analyzed. What do you mean by that? They studied different bacteria, different prokaryotes, and they noted that in most instances, the the Pribnow box consists of these 6 nucleotide sequences. Okay, so it is also commonly called negative 10 sequence because it is centered roughly 10 base pairs upstream from the transcription start site. Okay, and so this is another illustration of that, the Pribnow box. And what about this TTGACA? Well, it determines the frequency of initiation of the transcribed region of the gene. Somehow, it communicates with RNA polymerase bound to the Pribnow box. Okay? It will tell the RNA polymerase how many times will it transcribe this region of the gene. Okay? I think I mentioned this already. Okay, what else? So the Pribnow box is recognized by a subunit of RNA polymerase during initiation of transcription. Okay? And that happens to be the sigma subunit. Okay? I already mentioned that the promoter is AT rich. Okay? It is easier to break apart or unwind. In eukaryotes, this is the counterpart. Okay? The TATAAT box, the negative 32 box. Okay? And it confers fidelity of initiation. Okay? It tells exactly where the transcription start site is to the RNA polymerase. While the CAAT, GCGC, they are the regulatory signals. Okay? So again, like the TATAAT box, the TATAAT box is a sequence of DNA sequence that indicates where the genetic sequence can be read and decoded. The transcription is start site. Okay? It is a type of promoter sequence, like your Pribnow box. Okay? It is located 25 to 35 base pairs from the transcription start site. And it is the transcription factor that binds to the TATAAT box. Okay? There. What's the other name? Hugness box. Okay? Hugness box. Okay, I think I mentioned this already. It is a non-coding DNA sequence. Let's go to the next step, chain initiation. Now, this involves alignment of the first 2 nucleoside triphosphates to their complementary bases on the DNA template. For example, in the DNA template, you have you have adenine. Let's say, yes, you have thymine. Let's say you have thymine in the DNA template. Okay, what will be the nucleotide what will be the ribonucleotide that will form the 5' end of the RNA transcript. Again, you follow the base pairing rule. Thymine is paired with adenine. Okay? Now, if in the DNA template you have cytosine, what will you introduce at the 5' end? Or what will you incorporate at the 5' end? Guanosine monophosphate. Okay? Now, then the second NTPs follows. Let's say that the second base in the DNA template is adenine. Okay? Deoxyribonucleotide of adenine. What will you insert? Okay? What will you attach? I mean, you will attach uridine monophosphate following the base pairing rule in RNA. Okay? So the first RNA, the first NTP plus RNA polymerase forms the initiation complex. Then we go to chain elongation. Okay? The sigma factor dissociates from the core RNA polymerase. Okay? In order to expose the core enzyme. Now, it is very easy to remember chain elongation in transcription because it is very much the same as that in replication. Okay? The DNA template is read from 3' to 5' direction. Same. Direction of elongation of the RNA is from 5' to 3' direction. The same. And new nucleotide units are added to the 3' end of the growing RNA polymer. Same. Okay? So if you remember the elongation step in replication, it's pretty much the same as in transcription. And then termination and release. Sorry. The DNA template contains stop signals. It is ushered in by a region that is rich in guanine and cytosine, followed by a region that is rich in adenine and thymine. And there are two major termination strategies: row-dependent termination, row-independent termination, depending upon the species. In prokaryotes, in the bacteria, it is a row-dependent termination. Okay? So, row factor is a prokaryotic protein involved in termination of transcription. Okay? So, the raw protein tracks along behind, or it follows behind the RNA polymerase on the growing messenger RNA. And somehow, it helps in the recognition of the stop signals. This one. So interaction between the raw protein and the RNA polymerase, okay, releases the messenger RNA from the transcription bubble. So this is in eukaryotes. Okay? It is raw protein independent. And it copies the base sequence of the, what do you call that? Base sequence of the DNA. Okay? And then the RNA transcript becomes dissociated, and this is one option. Okay, one option. So what are the inhibitors of RNA synthesis? There are inhibitors that bind to the DNA template, such as actinomycin D, an antibiotic, aflatoxin. Okay, there are inhibitors that bind to the RNA polymerase, the anti-TB drug, rifampicin, and of course, amanitin. Okay, amanitin. And then lastly, for transcription, post-transcriptional modification of the RNA transcript, you have to protect the 3' end and the 5' end from the exonucleases found in the nucleus. How do you protect the 5' end? You attach a cap which is made up of methylated guanine nucleotide. How about the 3' end? How will you protect it from 3' exonuclease? By putting in a long stretch of poly-A nucleotides. We call that poly-A tail. Okay? We call that poly-A tail. That one. That is a cup made up of, methylated guanine residue. Then you can also modify the ribose through methylation. And it is important for the regulation of RNA stability, messenger splicing, and translation. You can also modify the bases. And that's the reason why the transfer RNA is different from messenger RNA and the ribosomal RNA because of the presence of unique bases, like so. Then removal of introns and splicing of exons. For the maturation of messenger RNA from heterogeneous nuclear RNA. And this post-transcriptional processing is important in the regulation of gene expression. Okay? Regulation of gene expression. We may have an overtime of 15 minutes. Okay? So of the 4,000 or so genes in the bacterial genome, okay, or 29,000 genes in the human genome, only a fraction are expressed at a given time. Okay? Only some are expressed at any given time. Differential expression of genes. Some gene products are present in very large amount. And what kind of genes and their protein products are constantly being synthesized? Okay? They are elongation factors for protein synthesis. Okay? So daily we synthesize protein. So we need elongation factors. On the other hand, other gene products are produced in smaller amounts, such as enzymes that are needed to repair rare DNA lesion. Okay? Common sense, right? The lesion is rare. Why do you have to produce so much of an enzyme? Then we have regulated expressions of genes that are required for adaptation. The need for enzymes and hormones in certain metabolic pathways may wax or wane as food sources change. Okay? So, of course, after meals, okay, after eating, you need a lot of digestive enzymes. Okay? For development, during development of multicellular organism from embryo to fetus, some proteins that influence cellular differentiation are present for just a brief period of time. Once they have done their function, the proteins are degraded. Okay? So, specialization of function. Okay? Specialization of function. And that's the reason why we need more globin in the red blood cell for the biosynthesis of hemoglobin. And there are genes that code for the alpha subunit and the beta subunit of the globin. So there are two types of gene regulation. Positive regulation. Okay? Positive regulation means you stimulate gene expression. You increase gene expression. Okay? Now, there is such a thing as double negative regulation that eventually result in positive regulation. So double negative has the effect of acting as a positive. Example, an effector that inhibits a negative modulator brings about a positive regulation. Okay? So at first, it acts as a negative modulator. Then you put in another modulator that will neutralize the effect of the negative modulator. So as a result, positive regulation, stimulation of gene transcription. And that process is what is known as derepression. Double negative derepression. Effect, positive regulation. Okay? So, negative regulator, negative effector will produce repression. And what is repression? Negative. Okay? Negative regulation. So there are three types of responses to a regulatory signal. Type A response. There is increased gene expression that is dependent upon continued presence of the inducing signal such as hormones, nutrients, or growth factors. Okay? So if you remove these inducers, gene expression will stop. Okay? So there is increased gene expression. Okay? Then you have the type B response. Increased gene expression that is transient, even in the continued presence of the regulatory signal. Example, response, desensitization, recovery effects of many pharmacologic agents. And this is the explanation for drug tolerance. Okay? So at first, you are very responsive, your disease is very responsive to the treatment, to the drug. But prolonged intake of the drug, okay, desensitizes the system. In other words, the drug is no longer taking effect. And then what will you do? Consult a doctor. And what will the doctor do? Change your drug. Okay? Change your drug. Okay? But later on, the doctor will bring back the drug because it is better than the substitute. And at that point, there is already recovery, response, desensitization, recovery effect. Lo and behold, the drug is effective again. The drug is effective again. Now, type C response, an increased extent of gene expression that persists indefinitely even after the removal of the regulatory signal. That means to say that after the removal of the regulatory factor, the effect persisted. Okay? The physiologic effect persisted. So these are the different processes involved in gene expression. And the most common site of regulation is gene transcription. Okay? Gene transcription, such as post-transcriptional modification, RNA stabilization. Okay? So this is the regulation of gene expression in bacteria. What is an operon? It is a linear array of genes involved in a metabolic pathway. Our example is lac operon and tryptophan operon, which is regulated by a single promoter region. A cistron is the smallest subunit of genetic expression. It codes for a subunit of an oligomeric protein. So one cistron, one subunit concept. And what is an oligomeric protein? A protein with two or more polypeptide chains, two or more subunits. Polycistronic messenger RNA is a single messenger RNA that encodes more than one separately translated protein. Okay? So you have only one messenger RNA, but within the single mRNA, you have several cistrons. Okay? You have several cistrons. And where do you find this kind of polycistronic messenger RNA? Bacteria, prokaryotes. How about we humans? What do we have? Monocistronic. Monocistronic. One. Another terms that you have to remember. Inducible gene. Okay? Genes whose expression increases in response to an inducer or activator. Without this inducer or without the activator, it has a low basal rate of transcription. Okay? What is a constitutive gene? One which is expressed at a constant rate and not subject to regulation. Okay? It is a gene that is constantly being transcribed into RNA and translated into a protein. Okay? It is always there. And that's the reason why it is called housekeeping gene. Okay? And inducible gene may undergo mutation and transform into a constitutive gene. And you call that constitutive mutation. Okay, let us discuss one, lac operon. These are the ones who discovered these two, Jacob and Monod. Lac operon describes how two adjacent genes involved in lactose metabolism were coordinately regulated by a genetic element located at one end of the gene cluster. So what are these genes? LacZ gene codes for a beta-galactosidase. This enzyme will hydrolyze lactose to galactose and glucose. So it is similar to lactase. It is practically lactase. Okay? LacY gene codes for permease that transform lactose into the cell. So these are the two that are very important. Well, this one is minor. It codes for a trans-acetylase. Okay? So what are the components of the Lac operon? Lac promoter, Lac operator. LacI encodes the repressor protein. And what is the inducer? Lactose. When lactose and glucose are present, the cell metabolizes first glucose. Okay, how does it work? First, without lactose or with lactose and glucose. LacI gene is constitutively expressed to form 4 repressor subunits that binds at the operator locus, thereby preventing transcription of LacZ, Y, and A genes by RNA polymerase. So LacI repressor is a negative regulator. There. This is the promoter site, and that is the operator site. Okay? And these are the transcribed region, Z, Y, and A gene. Okay? And what is this? The ribosome. Okay? So when the gene is expressed, it will form 4 protein subunits. That's the LacI gene. Okay? And this repressor is a negative regulator. The 4 subunits will combine to form a tetramer, and it will bind to the what? It will bind to the operator locus. Okay? Thereby inhibiting the transcription of Z, Y, and A gene. Now, this time around, with lactose and no glucose. So lactose binds, lactose molecules bind to the repressor subunit, thereby inactivating the repressor. So what happens? The repressor can no longer bind to the operator site. There. Okay? That's why LacI gene, that one, is a constitutive gene. It is constantly being what? It is constantly being expressed into RNA and RNA to a protein. What is this? That is a ribosome producing the repressor protein. Okay? Now, lactose, the inducer, will bind to the repressor. So the repressor cannot bind to the operator locus. So what will happen? The LacZ gene will now be exposed. Okay? How about in eukaryotes? Well, I mentioned some of them already earlier. Differential expression of genes. The beta-globin gene cluster is in the active chromatin in the reticulocyte, but in the inactive chromatin in the muscle cell. Okay? So even though we have the beta globin gene in the skeletal muscle, they are not being expressed. Unlike in the reticulocyte, the immature red blood cell. Okay? The beta globin gene is being expressed, and that is needed for the synthesis of what? Hemoglobin. Another is chromatin remodeling, such as acetylation of the lysine residues in histone. So what are histone? It is a component of the nucleosome. Wrapped around the histones is a segment of DNA. Okay? So what happens, and what is a histone? Histones made up of lysine and arginine, which are positively charged. What is the charge of the phosphate group? Negative. So what happens when the lysine becomes acetylated? It becomes less positive. Okay? There is a reduction in the positive charge. So there is decreased binding of the histone to the DNA, thereby disrupting the nucleosomal structure, now exposing the regulatory DNA elements in the promoter site of the gene. And then presence of enhancer and repressor elements. Okay, like this one. Okay, so what is this? Well, these are the regulatory elements present in the coding strand of the DNA, the one that is not being transcribed. Okay? And let's talk about transcription factors. They are regulatory proteins with special motifs that bind with high affinity to the correct region of the DNA. And the three unique motifs that are usually found in transcription factors are helix, turn helix, zinc finger, leucine zipper. So what do these motifs do when present in the regulatory protein? Well, they enhance the binding of the protein to the DNA. Okay? So it provides a ligand binding site for modulators or effectors. Because the modulators do not bind directly to the regulatory elements in the promoter site of the gene. Okay? So what binds to these regulatory elements? Transcription factors. And what binds to transcription factors? Modulators or effectors. Okay? Positive modulator, it will stimulate gene transcription. If it is a negative modulator or effector, it will inhibit gene transcription. And then gene amplification, increase in the number of genes available of transcription, increase in the number of RNA and proteins. And this is generated by a process of repeated initiation during synthesis, thereby providing multiple sites for gene expression. And this is the basis for the development of drug resistance, such as resistance to methotrexate, which is a chemotherapeutic agent and used for autoimmune diseases. Okay? So what happens? In the unamplified gene, there, you have only one gene. Okay? In the amplified gene, you have several copies of this. Okay? You have several copies of the gene. Then gene rearrangement. This is the process involved in the synthesis of different antibodies by lymphocyte. It allows the generation of millions of antibodies coming from a single gene. Okay? And this is due to what? Recombination. Okay? Of genes involved in the synthesis of the different parts of the antibody. The constant region and the variable region. Okay? So producing different kinds of antibodies. And then transposon, which we call jumping DNA. So what are transposons? They are mobile segments of DNA that move in a random manner from one site to another on the same or different chromosome. And this is mediated or catalyzed by transposase. Okay? And may cause diseases such as hemophilia and Duchenne muscular dystrophy. Okay? Also, exchange of plasmids that contain transposons containing antibiotic resistance or genes among bacterial cells may confer antibiotic resistance. And that ends our lecture. Okay? Last, methylation silences, okay, the gene. So transcriptionally active genes are less methylated. Okay? Transcriptionally active genes are present in the active chromatin. Methylation of the CG-rich regions may cause conversion of that chromatin into an inactive chromatin. So this suggests that DNA hypermethylation silences gene expression. And that finishes our lecture. Thank you for listening, class.